Hi, everyone. Thank you for joining us. We will be starting shortly. Hi, everyone. Thank you for joining us. I'm going to ask the panelists to turn their video cameras on for us. And we are going to do some introductions as we go along. Thank you for joining us. We greatly appreciate you being here today. I see that we still have some people filtering in, and we have a great amount of people here, and we are so excited. So welcome, I'm Stephanie Aronson. I'm the Community Development Manager at the Strome Jewish Community Center.
And I'm Nomi Mitchell, the Interim J-Connect Manager at Hillel University of Washington.
We are so happy to be offering this experience tonight. We are lucky to have someone who has dedicated their career to vaccine and immunology studies to speak with us. The rapid development of the COVID-19 vaccine has been interesting to watch unfold, and I'm excited to learn about the process of vaccine development and trials as this comes to market. Before we continue introductions, as questions come up, please put them in the Q&A chat. Nomi and I will be back on at the end to ask your questions of Amy and Dr. Corey. And now we are going to make introductions of Dr. Larry Corey and Amy Lavin, who will take you through the next hour.
Hi, everyone. Dr. Larry Corey is an internationally renowned expert in virology, immunology, and vaccine development, and the former president and director of the Fred Hutch Cancer Research Center. His research focuses on herpes virus, HIV, and the novel coronavirus. Under his leadership, the HIV Vaccine Trials Network has become the model for global collaborative research. He is also the principal investigator of the Fred Hutch-based Operations Center of the COVID-19 Prevention Network and co-leads the network's COVID-19 vaccine testing pipeline. He is one of the most highly cited biomedical researchers in the last 20 years and is the author, co-author, and editor of over 1,000 scientific publications.
Amy Lavin now serves as the CEO of the Strome Jewish Community Center. Just prior to that, however, she spent 4.5 years working at Fred Hutch, creating and building Obliteride, an annual event that raises money for the amazing research happening at Fred Hutch. Amy, you're going to take it away in a moment. Nomi and I are going to turn off our videos, and we will be here looking at the Q&A, and we'll be back at the end.
Well, thank you both.
Pleasure to be here.
Nice to see you. And
Okay.
thank you everyone for being here. We've been looking forward to this evening for a long time. As was just mentioned, I had the great opportunity to work with Dr. Larry Corey for years at Fred Hutch, and I continue to be a huge supporter of the Hutch's work and Larry's work in particular. And so tonight, we're excited to bring to the community greater understanding and insight into the vaccine development process and just how Larry's lab at Fred Hutch is contributing to what's happening globally and also what we'll be able to benefit from locally in the coming months, or hopefully months and not years, but there's a lot ahead. And so thank you very much for being here. We are very happy to have you. And everyone should know, if there's two things I love, it's working in the Jewish community and Fred Hutch research. So this is a great combination.
Well, this is sort of fun for me because, you know, I've been admiring Amy since her bat mitzvah. So, she and it was fun to recruit her to the Hutch, and it's fun to do this tonight.
Well,
so
thank you. And I'm glad we
the
could be
object
here
is,
today.
we'll have some fun here tonight.
All right, great.
All right.
So, you know, I read as much as I can on what's going on around us, how quickly our lives are going to revert to something or move into a new type of normal. And so for those who follow this, maybe not as closely as you obviously do, and those of us who are reading daily, if we were just to start by getting the key question out off of everyone's mind, which is how quickly do we really think life will, you know, tend towards something that looks more normal, where we can spend time with our friends and family with a bit fewer restrictions. So just a little bit about that, and then we'll get into the vaccine development.
All right. Well, you know, the reality is that we definitely need science to lead us out of this. You know, you'll see these vaccines are, you know, better than I expected them to be, you know, back when I started this journey in, I guess, March. But, you know, there are a few things we have to understand. The vaccines, there's still vaccine scarcity, and that's the hard issue to deal with. Is it's still a few months before we get everybody fully immunized. I'm happy to be able to, you know, I'll say a few words, give you some background, and then be there to answer questions. The vaccines give great personal benefit. We actually don't know if they actually are shifting the spectrum so that they you don't get sick from it. They're fantastic. They essentially, the RNA vaccines are 100% protective from hospitalization. So they're going to be of great personal benefit, and any of people of my generation, I'll say, you know, I sort of can't wait to get vaccinated. So when that happens, and it's offered to me, I definitely will accept it without any issues with respect to the risk and the benefit. I'll outline that in a couple slides, I think. But yet, you know, there's still some mask wearing, because we actually don't know if you could still get infected, not be symptomatic, still be infectious to others. And we're still left with the issue of if someone comes to dinner or your house, do you know they're vaccinated or not? And could you still transmit it? And, you know, in stranger situations, masks are still going to be around. I'm in the midst of designing a study to show whether that can happen or not. But, you know, if we crank it out and get it done by the end of the spring, maybe we'll know that answer. But, you know, the reality is that if we get enough coverage, enough personal benefit, I think we will see some element of normalcy. Normalcy for a lot of people is going to be getting their kids back to school. And so the pediatric program is just starting. You know, RNA vaccines, I'm not afraid of it for kids, but I prefer some of the other platforms that I'll talk about for my grandkids. That will still, the pediatric program just starting in January. And, you know, I'm optimistic that we should be able to plow through that. So by the end of the summer, you know, for fall, we should have a chance of having everybody vaccinated and you know, some element of life, normalcy. You know, there are lots of things that go on with economic recovery and trust and how we actually get ourselves back to, quote, where we were. But, you know, we know that it's still going to be, in a lot of things, it's still combination prevention. Vaccine is going to be incredibly great. But there's not one thing always that's going to be a silver bullet. Well, we haven't answered all the questions, and we'll have to see.
But I should take a minute to note that the timeline at which, the speed at which this has been explored, developed, and now I just want you to talk a bit about, give some people some perspective. And I know we hear about it in the news, but as somebody who's been so close to this type of work and research for decades, like, how does this compare?
It's unbelievable as to what science has delivered and what American science has delivered. You have a virus that's just discovered. The gene is, put up on a website on January 8th. Even here, we have a diagnostic test in the virology lab at the UW within, with a week. It took the FDA, we couldn't use it for about a month and a half until the FDA, approved it. Yeah. But the Vaccine Research Center, which was started for HIV vaccines 20 years ago, it had then been working on SARS in the past, the SARS-CoV-1 and respiratory syncytial virus, kids virus, and it identified that the best antibodies were when the virus had sort of sat like this in an open area, and in what we call the prefusion complex, the open complex, when it lands, it then would close. But antibodies to the open state were really the best antibodies, and they learned how to stabilize them 5 or 8 years ago with SARS-CoV-1. So within 3 weeks of the gene, they had actually figured out what the crystal structure was, and that this structure was the way it looked, and put it into animals and gave great antibody, better than others. And then they transferred that gene to the companies, and 5 out of the 6 companies are using this, and both RNAs basically are the same vaccine. So that science building on science, the fact that you knew the crystal structure in a month, you then start making a vaccine, you get it into humans, by March, or by April, May, and then we crank out 2 30,000 person trials. Well, one at 42,000, one 30,000, and essentially by Thanksgiving, that this stuff works. I mean, this is just unbelievable that in 11 months this happened.
That's amazing. So tell me, if you can rewind a little bit to thinking about someone who's been in your field for a long time. You don't know when something like this is going to land. So, and you've obviously explored various viruses over your career, but when you saw this emerging last December, January, did you, what were your initial reactions about attacking this from a, research perspective, and also just like, what is the world in for?
So, at first you read the newspaper and you look at it, and then you start understanding that this is a virus that has staying power. And, I fly to DC about once a month, and I have, in the HIV business, we have a routine. I go to Tony's office at 6 o'clock. We talk for a half hour. We go out to the same restaurant. We go to dinner, and we talk and sort of do that. And in January, we both sort of knew that this virus had staying power. There was so much going on in Wuhan that you sort of knew that this was not going to be something that burnt out. You saw such, adaptation to human infectivity. So that, so you start paying attention to it. And I mean, I think I largely ignored it till I came back from, until February. And then it had started in Europe, and at that point in time, it was really apparent that we really had something here that was going to be significant. And of course, it came to Seattle first, and our city handled it as well as any city had, because we had built resources of a lot of people, frankly, who I had trained in the virology lab, and we had a lot of virologists, and we had a lot of influence, and we were able to set up a testing program and sort of do things better than other cities. So okay, that doesn't mean we're out of the woods. And it became very early apparent that a vaccine was going to, quote, be useful. Probably didn't quite understand how. In fact, I don't think anybody could predict, even myself, that how extensive and how infectious this virus was. Subsequently, we now know that you can take a baby hamster and infect it and put another little hamster next to them, and only one infection, and watch them breathe, and you separate in cages, and it takes only one infectious unit to infect that other little hamster, and it takes 12 times as much virus than flu. So this is not flu. This is A, more infectious actually than flu, and B, more dangerous than flu. That formidable issue that comes out later in time.
Mm-hmm.
But you recognize that, gee, it's time to pivot. We have a crisis here, and how do you solve that crisis? So science builds upon itself, and use the resources that you have. And we have built a lot of resources in HIV and influenza that we could essentially take the medical infrastructure that we had, all the medical schools. And so when when Tony sort of approached me and sort of said, hey, we need to pivot and want you to do this, it wasn't that hard to start calling all the friends I've had all these years in the infectious disease community that all been taking care of people in their hospitals, and to sort of say, hey, will you direct some of your efforts and your staff to building an army of clinical investigators at the medical schools to test candidate COVID vaccines. So those were easy phone calls. 99 out of 100 said yes. So you sort of build this army, and the large companies were making the vaccines. We wanted that to happen because we actually know we needed multiple vaccines. It was very clear all the adults need to be vaccinated, and become very clear now all the kids are vaccinated. So you can't travel unless other people are vaccinated. So this was a problem of major scale. We had 330 million people in our country, and we got 7 billion people on Earth. And frankly, if the economies are going to work, and you want to go, you want to go to Italy, or you want to go to Cambodia, or wherever you want to go, everybody needs to be protected. It is a global problem. It's not really America first here. It is really the issue that if we're going to be protected, everybody has to be protected. The virus clearly tells you that it's not black, brown, red, blue. It is everybody. And density and human contact are its friends.
So I'd like to understand, or if you could share with us, kind of two different dimensions of the vaccine development. One is thinking a little bit about how vaccines work and how these recent vaccines are both similar and different from past vaccines for prior diseases. And then also, we can get back to it if you don't get there, but talking a little bit about all the infrastructure that's been sitting in the trial networks that you've built, and a little bit about how you were able to pivot those quickly, not only with the army of people you rallied, but with the trial network for community engagement.
Well, how vaccines work, I got some colorful slides. They were once shown to, once shown to Vice President Pence, so maybe they'll be fun to look at. Where'd it go? It was here. Maybe we're not going to show these slides. I'm sorry. They were supposed to be on my desktop. All right.
Go ahead and talk to it. You're familiar with the content.
I am embarrassed to sort of say that here it is. Let me see. Let me try again. Share screen. And even though it says Hillel presentation, it's not coming up. All right, I'm going to talk to it.
Perfect.
Okay, we'll just forget it. Vaccines work by, you create something that looks like the virus, that's not the virus. So the mRNA vaccines means that this coding region of the part of the virus that lands on a cell. So it's basically, the concept is if you block landing of a virus, and I had a cute slide of the lunar landing. If you block the landing gear, and it essentially will not land on your nose or your lungs, or the various other places where the receptor of this virus is. So vaccines take the structure of the virus as part of the virus. You don't have the virus itself. Actually, with these RNA vaccines, you actually take the RNA, DNA makes RNA, makes protein. So this is the RNA that is the code that transcribes the protein. So it has the open structure of the landing gear. And then you take that, because we have things in our body that degrade RNA, because having RNA around that transcribes things is not something that we like. So you actually put it in a bunch of particles. It's actually like soap particles. And that is actually what the RNA vaccine is. You inject it, it goes into cells. And as soon as the RNA, the lipid particle is engulfed by the cells, and the RNA gets into the cell and it starts making this structure of the landing gear. And that actually is presented to the body, and you make antibodies. So you make those antibodies. And so the reality is you make those antibodies, they get put into what we call memory. And then if you get exposed to that virus, that cell that is in memory will say, oh, that's my job. I'm going to go there and make some antibodies to that stuff. I'm going to wake up and do this. And that takes about a day or a day and a half. If you don't have those antibodies in the first place, it takes about 2 weeks to start making antibodies. During that period of time, the virus is a happy little critter, and it'll replicate about, once every 30 or 40 minutes, so it doubles all the time, and you get it in the body. That's what makes you sick. So the antibody response actually, prevents all that from happening. So that's basically how a vaccine works, and that's how an RNA vaccine works. So that's sort of in a simplistic form how that happens.
And I think I've read that this is different than prior vaccines. So maybe just a little bit at a high level how it varies from others.
This is a great demonstration of the sort of the power of these new technologies. So you can make RNA in the lab for a couple $100 in 24 hours. We call it paper cloning. You write down the gene sequence, you send it to a company, they actually synthesize it in a bacteria, and they send it back to you in about 3 or 4 days for about $150. So you can just do this.
I can order that online.
You order that online. You don't even have to do an experiment. It just sort of comes to you online.
Yeah.
It's like Amazon shopping for scientists.
Exactly.
And so you can do that really quickly. There's another new technology that we have, and that's personified by the Chimp Adenovirus vaccine, or the J&J vaccine. And that's sort of using a disabled virus, and you drop the gene. This time, usually it's the DNA of the gene, the same gene that you drop into there. And the virus sort of shuttles it into, you inject it, and the virus shuttles it into the muscle cell. So the virus is a shuttle vector, just like the space shuttle, and it carries it into the cell. And the same thing happens. This time it's DNA that makes the cells, that takes over the cells. The cells makes the RNA, and then it makes the protein. So actually, the Janssen vaccine is the exact same gene as the Moderna and the Pfizer gene. And the most of us get vaccinated is you actually make the protein. Now that's, that actually turns out to take more time. It's more skilled. It's folded all over the place. It's in the right configurations, and you have to get that just right. Harder to make, but once you make it and you set it up, and you can, the big factories have already been made, and you can actually make a lot of it cheaper than this newer technology, which is quicker, but a little bit more, substantially at the moment more expensive. So that's how most of our vaccines are. Vaccines for hepatitis B, almost all the kids' vaccines are these kinds of viruses in which or vaccines in which you actually make the protein. And those actually, and this worked out just this way. The RNA came first. The viral vectors are in clinical trials. They were made second, and the proteins were just finally getting into clinical trials this month and next month. Frankly, those are the vaccines that, from a platform point of view, pregnant women and kids have traditionally had, and the ones that we tend to use in children. And so, and when we designed this program, we knew we needed, it was really like Lake Wobegon. We wanted all these children to be above average. We wanted all of them to work, because not one of them can be for the entire country. This whole issue of vaccine scarcity been known to us since we started the program. When you sort of see the country today got 100 million doses of this or that, it was, as much as I love the New York Times, and but the article today bothered me that the government didn't buy, had the opportunity. Well, would you have bought, when they were charging 5 times more than anybody else in July, and you had no idea whether it was working, and Moderna was being charged less, would you really have bought $400 million of 400 million doses at 10 times higher dose price than anybody else when you didn't know it worked?
Right.
Now today, hindsight's 2020. You can sort of say, gee, it would have been better if we bought this from Pfizer, but it's not like they were giving it away, and it wasn't like they were actually absolvable in this. There's nothing wrong with Pfizer. I'm not anti-Pfizer. I'm just sort of saying that
you were placing a bet at the time, and it was, are you going to place this bet? Yeah.
And you didn't know how well it would be. And the whole program is that, each of these vaccines had a chance to work because they were looking at the same gene. We now are pretty sure they'll all work.
That's amazing.
We have the monoclonal antibodies, which, Trump got, that they work, and that's against the spike and the target. That's all science building on science, saying, we picked the right thing to go after. This virus is a lot easier than HIV. It doesn't mutate like HIV. It's structurally pretty, all the mutations that they occur are about 2 a month. HIV is like 2 a minute. And so we are able to have these vaccines. I think they'll last for a while. And, science worked. We picked the right target, and it looks like it's working, incredibly well.
That's amazing. And I think once you start to learn more about this field, you realize the intelligence of these viruses themselves, and those that mutate faster. I mean, keeping up with them, it's a fascinating element of science. So, and I'm sure your experience from HIV and everything else before that has been helpful. So how else, when you think about Fred Hutch's role, and I very early on saw that you were leading some of this nationally integrated trial work. What, does that look like computers? Does that look like, lab work? I mean, just for those people who don't get to see that, it's kind of hard to see what's inside the science at this kind of stage in a fight.
Operations are, there's a lot of people working at their desk on computers and sending emails and being on the telephone. And I'm not, there are, when you're working in the lab and measuring, what's the antibody levels, and there's a lot of plastic and a lot of pipettes, and people have seen all this. I mean, science is part of science is still working with your hands, actually, to grind out what are these, what experiments look like, and there's fancy machines and simple machines. But in the end, it's about people, and doing a clinical trial is about getting people into the clinical trial, and basing altruism of people as it relates to sort of their role in society. And we have a lot of public relations people. We have a lot of people with community organizers. We've been working with disadvantaged communities for years, building trust, trying to get Black and brown people who have suffered inordinately. Unfortunately, the slides don't show this. I have some really, the data are startling. If you're Hispanic or Black and you're under 65, you have a 7 times greater chance to die from COVID-19 than anybody who's white. And it sort of changes, that disparity changes only if you're, 75 and older, where the death rate is equal. So density is the issue. It's not genetic. We see the same data in the UK and the same data out of Europe. So it is the exposure rate. It is the essential services. It is being in contact with people, not having resources to have the kind of house that I have here, with not a lot of density. So a lot of this is really community organization, and the medical schools of the country tend to be in the area where, those are the catchment areas, and they've worked with these communities, and that is how we've mobilized people to be in the clinical trials. And we've had an unbelievable epidemic in the country, and frankly, the clinical trials are based on the number of cases that you have. And we planned the clinical trials from beginning to end to be 6 months. We sort of said, well, the statisticians came to me and said, 15,000 per trial is all we need. And I said, well, we need the answer as quick as we can. So we doubled the trials. It's 30,000. That's what we're going to do. So we planned that from beginning to end, it would be 6 months. It ended up from beginning to end 4 months. The Moderna trial, the Pfizer trial, they started on July 27th.
Amazing.
And by November 15th, we had enrolled 30,000 people and had the number of, quote, cases to discern this amazing difference. And the fact that both trials run independently, sort of with different populations, come up with identical results is like just this, to me, is just astonishing. I mean, it's astonishing from two ways. Number one is that the data are so good and they're so similar, but astonishing also that you take this one strand of RNA and you wrap it in a different lipid and you inject it into people's arms, 30,000 different people. I mean, 15,000 in one trial and 22,000 in the other trial. So it's, 47,000, and you get the same result.
It's amazing. And is there anything that's actually been surprising a lot, besides the fact that it's been so successful? Have you learned anything else about the process or human, like body anatomy, anatomical reaction, anything else that's been surprising, or you guys really had a handle on it and went for the right thing?
Oh, I can't say you have a handle on it. I mean, people are always asking me about the side effects of the vaccine and things like that. I mean, the side effects of, if a lot of you have taken the Shingrix vaccine, the zoster vaccine. So it's a little bit better than that. It's interesting that the younger you are, the more reactions you have. The second dose gives you a little bit more than the first dose, but it's very well tolerated. I mean, with the second dose, take a couple ibuprofen or take a couple acetaminophen and you'll be fine.
Great.
But the risk benefit is, it will, the protection from being hospitalized, developing low oxygenation and having a heart attack or a pulmonary embolus or all those other things that actually happen with this disease, is just markedly mitigated.
Amazing. And are you working on a global network right now? Or how connected internationally is this whole effort?
Yeah, I mean, under the Operation Warp Speed, the program has always been global. In my mind, it's always been global. Now, the first global vaccine we've been doing is the Rna Vaccine, where over half the people enrolled are internationally, and that goes from Africa to Central America. Pfizer did a few people in Brazil, so and the protein vaccines are half the study is sort of offshore and half of it's inshore. So, as I said, the program has always been global. We've always worked with the multinational big drug companies because you knew that the goal is to get it into people's arms as quickly as you can. So working with small companies is really problematic. And also you wanted the global companies because they have their global distribution networks. That's their business.
Well, great. I think the other thing maybe that we could talk about is, I believe the Hutch's role in some of this stuff has shifted from coordination and administration. And I believe I read that in this next phase, maybe a phase 3 of something, the Hutch would actually be managing some of the actual clinical execution here, or the trial execution with our own local community members. Is that?
Yeah, there are 3 clinical trial sites here. And then the Hutch's, we've also built a special clinic for COVID positive people to, so the experimental monoclonal antibody therapies and some of the new antivirals, are set up to be here. So the university and the Hutchinson have always been melded together in the infectious disease division as well as in the cancer area. So the city is blessed with some really, the medical care in the city is really terrific, and the research establishments are really first rate. So we're at the forefront, both from a therapy and a vaccine point of view. So, okay.
Wonderful. And I think I just want to underscore that how fortunate we are to have the Hutch here in Seattle, and paired with the partners in the network and the alliance that serves our cancer care community so well, and all your work that obviously has been designed and serving so many purposes over the decade is a gift to our community and the world. So thank you for all of that. I'm wondering if we have any questions that we want to tee up right now. If not, I've got a couple more.
Yeah, no, it's.
I'm seeing one here, and I don't know if Stephanie and Nomi.
Again, thank you. This is so interesting, and I love seeing the questions come in. I can see people's minds marinating and moving with it. You were just talking about a few different pieces and steps, looking back, looking forward, and a few people did note how might this and your experience here impact different studies and trials moving forward in different areas, or what lessons have you learned that may translate differently forward?
Well, I think the major issue is that when there's a will, there's a way. I mean, what I love about Operation Warp Speed, although it's affected my life, I'm not sure Amy loves it, but usually when you have something, you'll say, gee, let's talk in a week and we'll do this. With Operation Warp Speed, it was like, well, let's talk tomorrow. And it didn't matter if tomorrow was Saturday or Sunday. And I have to say, since March, that's the way it's been with me. I mean, I start my day. Only thing different about Sunday or Saturday is that it starts at 7 rather than at 5:30 in the morning, and that's just, and so when you have a problem, you can solve it when you agree that everybody can work together. When academia works together and industry works together. And another issue is when you're given essentially an unlimited amount of money. And that has been the case here. We have really gotten all the resources one needed to do this and do it well. So when the country wants to solve a problem, I think this is actually something that you can solve. Now, I think some of the problems that are non-biology, can be solved when there's a will, there's a way, and you have common good. And we don't ask each questions about our thoughts on something. It's really on whatever it takes to solve this problem. Let's sit and collectively try and solve it. And I hope that will go to other diseases and that the way of doing things, not that everybody can sustain. You certainly don't want to sustain working at home and sustain the kind of pressure that one is on with respect to work-life balance when you don't have your kids in school and things like that. But there are some things that can be learned about this sort of team, about the pace of discovery and the acceptance of trying to get to yes as quickly as possible.
So one question I'm also seeing pop up is both about the reoccurrence when we're seeing people relapse and get it again, and they've been very rare cases as I've read, but they might be there. And how might the vaccine, I've seen a couple questions pop around that, and also, will people who have already received or already had COVID-19 and theoretically recovered need to get vaccinated?
All right. Point one, this virus is not going to go away. It's not ever going to go away for years. And we're a long ways away from what people call herd immunity. Even you go to the Midwest of the United States, it's maybe 20 or 25%. Here, it's still 15%. That means 85% of us still haven't gotten it. So the fuel for the virus is there, and it's incredibly infectious. It's not influenced by seasonality, really. Look at what's happened in the summer, and then you go indoors, it's even more. It's incredibly well adapted to our noses, and it's subclinical, so you don't know you have it, and So, we can all get vaccinated, and hopefully it'll have some public health benefit also. And when that level occurs, but I think that's sort of years away. But in comfort is that I think both the vaccine and a natural infection, there are studies to say that even though antibodies wane, that's normal, but you put into memory, most people put into memory the antibody kinds of cells that produce very good neutralizing antibodies. All right, so therefore, when you see that virus again, or we see this virus again, you will mount, say with 95% assurity, you will mount what we call an anamnestic, a quick response that I said that will, block this virus. And I don't think with either natural infection, and I'm pretty certain with vaccination, you will see a prompt response so that it will be a milder illness. You're not going to get that response that, takes you 2 weeks and the virus is replicating and you end up in the ICU because you have a, severe pneumonia and you have virus throughout your entire body. So I do think it'll be much more like flu or the common cold or what the common other coronaviruses are, which is you get these coronaviruses every couple of years. You get, you make immunity and it goes away. Now, I think the immunity you make with this virus, SARS-CoV-2, is way more than a human cold virus because this is really much more of a systemic illness, and it really will disseminate throughout your body, and you make stronger immune responses with systemic illnesses than you do with just, respiratory illnesses. So that's a very long-winded way to say, yeah, we're going to see it, we're going to get it, but I think, yeah, through vaccination or having it, you're going to do a hell of a lot better.
Great, thank you. Go ahead, Stephanie, if you have one.
Thank you. Yeah, and I see, and I love this question of people asking how they can continue being educated. And I know it's something I personally feel as well. How do you suggest we continue after this conversation to best understand the vaccine development? Where should we be looking? What sites should we be keeping on tab?
I do think this is going to require both patience and sort of some understanding that we're all in this as a society together. Like I say, I think it's really hard to cope with vaccine scarcity. To know that this stuff is not there, and what is going to be the distribution system? I don't even know. I mean, I know some, first responders and medical care workers, but then how do we balance the elderly versus those who are in essential services? The elderly is 90 million people. This people in essential services is 90 million people, and all the healthcare workers are 24 million. So that's 200 million. We have 200 million doses, which means 100 million people. So that's 50%. Now, we're hoping that in January, the J&J vaccine and maybe the Chimp Adeno vaccines will come available, and they add by April another 70 million doses. Just the J&J vaccine, the Chimp Ad maybe another 70 million. So it may require other vaccines, just an RNA, to get everybody vaccinated the first time. And I think that's going to be hard for us. I think that, and how it gets distributed and where it will be, I think there will be a balance. There needs to be a balance between how this could be.
So there are also some questions about kind of the point of delivery, both the pricing of something like a vaccine, which I think I've read is supposed to be free to people, And also some discussion about, will it be potentially distributed through schools or non-traditional sites? Maybe, those of us who go to flu shots, we go to obviously the drugstores, even Costco this year had it, but what do we see as distribution points?
Well, the vaccine has been bought by the US government, so no one should be paying for the vaccine. And as far as the delivery charge, Walgreens or CVS, if you get it that way, going to charge you or not. Probably if you have health insurance, they'll charge you, and the health insurance policy, I think, has been covered in the CARES Act, that they have to pay for that. And then there'll be public funding, I'm sure, they'll extend the child's vaccine program to adults. And so I think that money is not going to be an issue here with respect to access. As to how it's going to get distributed, there's no national program here. There's 50 states programs. It's sort of like the Electoral College. And I have confidence. I think the state health department and the executive branch have done a reasonable job. And, but I think the allocation that comes here and what county it's going to, and then how's it going to go there. And, the Pfizer vaccine's packaged so that you pop open this thing that has 972 vials, and they have to be vaccinated in a few hours, frankly, 6 to 8 hours. So it seems like that should be distributed at schools, and you go to the place and you get vaccinated, and a lot of people, like the old days of polio, et cetera.
That's what people were referring to. Exactly. Yeah. That was the exact reference.
The Moderna vaccine is packaged in 5 doses per vial, so that's more amenable. As I'm gonna walk into Walgreens or CVS and see if I get an appointment. And how do I get an appointment? Who contacts me? I'm clueless, actually. And I'm actually not sure how anybody finds you and makes you eligible. I hope it's equitable. That's where I hope it's
obviously be a national objective. So that's great. The polio thing was the exact reference someone was making, in that it was done through schools so that everyone could have access as swiftly as possible and equitably as possible. So Stephanie, I think you had another one.
I'm actually going to pop in if that's okay. There was a question that came in about the anti-vaccine movement and whether or not you think this movement has grown this year, and for the parents out there who might not want their kids to have a vaccine in the coming years, what can we expect for kids today and in the future?
Yeah. What can I say to this? It's disconcerting to me, but people will believe what they will believe. And I think in some respects, I can only at times shrug your shoulders and say, we agree to disagree, and you have control what you have control. I'm not someone who believes at the moment that we should have mandatory vaccination of these, because I actually think it creates more disincentive to not take the vaccine. I can't say I've ever seen a vaccine that has more personal benefit than this particular vaccine. We have a disease that is affecting all of us, affecting our lives, about how do we live, let alone the hospitalization issues. And the most important disease aspect of it is that there were 30 severe cases, which means by definition hospital cases, in the Moderna trial, and there was all 30 were in the placebo group with 1 death, and 0 were in the vaccine group. I mean, so I'm looking at that saying, whoa. I just wrote a blog saying, keeping these people on placebo is wrong, and vaccinating everybody in the placebo group with a vaccine is what they deserve. And it just shouldn't be the medical workers, because the endpoints were really in the Black and Hispanic essential workers. And, so everybody is a band of brothers in this, and sisters in this cohort vaccine study, and they all helped each other, and they all deserve the same, response. So the placebo recipients deserve to get on the vaccine, to get the vaccine. So I think this vaccine has incredible personal benefit. and will say that, both my daughter-in-laws are physicians and are out there on the front line. I'm saying, you absolutely get this vaccine. That's the way I feel about it. I think the data are incredibly precise and good, that it is a great personal benefit. The risk-benefit ratio is very high.
Yeah. And can you reiterate for us, because we're getting a few questions, just reiterate, if you get the vaccine, what is the likelihood of you spreading the virus to others if you're traveling or seeing people after getting it?
Well, the answer is at this point in time, I don't know. Okay. We designed this trial and these trials to prevent the disease. And there was a lot of reasons for that. That's the way most respiratory viruses are. It was sort of a consensus that we might not be able to prevent you from acquiring it at all, but we could prevent you from progressing to disease. And it clearly does that. So the question that I'm being asked now by everybody is, gee, can I still get infected? Don't know I'm get infected and transmit it. I mean, the White House events and all that stuff, everybody knows asymptomatic carrier is how you mostly get it. They don't know they have it, and you transmit it to others. Am I gonna be like that? I'll be fine by myself, but I'm gonna be still at risk to transmitting it to others. And the answer is we don't know that. And we're trying to answer that. And what's the corollary of me saying we don't know? It means you still gotta use your mask. You still gotta have physical distancing. You can feel way more assured that, if you get it, you're not gonna get it. It's not gonna hurt you, but will you be still able to be the vector for somebody else, or someone who you love, or someone who you're in contact with who hasn't gotten vaccinated yet? And so we're going to have that uncertainty principle until we answer that question, or we get everybody vaccinated. Well, we know we're not going to get everybody vaccinated by, even by the fall. We're getting close, but and so we still have some uncertainty. It's still not, gee, we've turned the clock on and we're all better.
Yeah, and I appreciate you answering like that, and it leads right into that previous question about how do we follow along in an informed way to make sure that we're all staying up to date and keeping our loved ones and community safe. So thank you. Another question that's coming in is about frequency and boosters that might be needed. Is there talks of that within how often people might have to go back and receive a vaccine?
I don't know yet. It's a delightful problem to be able to talk about. All right, we answer first questions first, and we'll see what happens. It is the reason why we are following these people. We're in the first trial for up to 2 years. We can look at those who got vaccine first and placebo second, and placebo first, and they got vaccinated about 7 months later. We'll follow both of them. We continue to follow them and see if there's a drop off in vaccine efficacy. Is there a drop off in antibodies? And I'm sure they'll be boostable. But so we just don't know yet.
And that's very interesting, because I think some science, your point about science building on science, some of that happens very quickly and some of that takes a while. And I know that as I was closer to the science at the Hutch, when I was there, your career is one where you've got these long horizons and you may not see the payoff for a while. So, I mean, so what's happened in the past year is remarkable.
Yeah, I mean, that's a question when people would ask me, I'd say, gee, I'd love to be able to, I want to answer that question, because it meant that it was worthwhile getting in the first.
Boost.
All right. So if I'm worrying about a booster, it means, oh, okay.
Very good shape. Fair. So a question is popping up about the different requirements for refrigeration. And many of us have heard this, that I believe it's the Pfizer one requires an incredibly cold transport and storage. And so just a little bit about what's causing that distinction, how that impacts distribution, et cetera. And you talked a little bit about it before, but I think there's some more questions.
I've always felt that was General Perna's problem, not my problem. He's the freezer guy. Anyway, at the moment, the Pfizer vaccine has not been done in a way that they've done the tests to show that they can, take it up from a -80 storage where you preserve everything at dry ice. So it's being transported in dry ice, and then for a few hours it can be at it thaws, and it's good for whatever. So I'm sure they're making some adjustments that will allow the stability test to see that the stuff is stable, but that's really the issue at the moment. It sort of complicates this with respect to why the vaccine is mainly being used in it has been designed basically for Europe and the United States. It's very difficult to conceive that you're going to be able to do this in sub-Saharan Africa, and it's why the other vaccines are so important globally.
And I'll also add, I know you have a lot of experience as well in working in Africa and understanding how different geographies call on different types of activities. So we're getting a question if you can define the range of elderly, which I don't know if anyone can define the range of elderly.
The range of elderly
of this.
Well, elderly gets older and older each year
As
with
you
me.
get older, yeah.
So, but the data as cut by the CDC, where you really see an increase up in death rate, is 65 and older, especially 70 in every decade there. But the vaccines have been cut at 55 and older, and so. But I think the distribution is. It really is by decade, and you saw great evidence of that in Britain. How Britain handled it with the Pfizer vaccine is, they were first going with everybody over 80, and they first did the 90-year-olds before they did the 80-year-olds. So that's how they're working it down that way. I think we're going to be a little bit more egalitarian in how we're defining the elderly.
Yeah, fair. So one more question I'm seeing is, I think as this is such a multifaceted problem, right? You've got obviously the research going on, and obviously we've all heard about how at the same time the production has been done so that it can be distributed and activated as swiftly as possible. Some questions are up there about, do you have any concerns about what could go wrong? Obviously, refrigeration is one that you don't want to waste a single vial, but any other concerns that we should, that we might hear about, or things you guys are working to avoid?
Well, my biggest hope is that we don't waste any vials, just as you pointed it out. But I think also expectations. Manufacturing vaccines and biologics is tough, especially also under pressure. So, having writing the paper that the filling aspect of the vials were bad, or this were bad, and 1 million doses were lost, and this kind of stuff, or the truck got lost and this happened, the stuff unthawed. This kind of stuff happens. It's like the bad delivery. And
the snow could stop the truck.
Yeah. And so we're going to read about it. And I guess I'll say, all of us try and have some equanimity, that there's a lot of people working who are, very smart, very hardworking, but getting this perfect is not going to be easy.
A question that came in, taking us in a little bit of a different direction into politics. What changes do you think we will see in 2021 with the new incoming government as far as the COVID narrative and what is being put out to the country?
Well, I think we're going to see it right away. Is the this much more uniform, national guideline that we need to wear masks, and they are really quite effective. And that physical distancing is there, and being indoors with a lot of people, and understand that there was a reluctance even on scientists to recognize that it really was aerosol and not just particles. So real early, we all saw the MIT thing when you sneezed and you saw the dye all over the person, and that God, is that really, but no one actually did the same experiment for months. They did it, but they didn't really illustrate that when you talk, you are actually aerosolizing. And those particles don't go as far, but they go. And so when everybody is in a restaurant and talking, And the smaller particles actually, because they're lighter, don't fall to the ground. They are suspended more. That is, the virus is there. So if you get 50 people talking in a small room in an unventilated bar, and you're all animated, and you are drinking alcohol, and you talk more, and you're louder, and you're keeping your mouth open more, this is, wait a minute. This is the ideal way that we design experiments to aerosolize stuff when you want to show that it aerosolizes. And so that has to be recognized, that is not safe. And okay, there's an economic aspect of that, and that we don't cope with that. And we don't handle, I mean, in Great Britain, I don't know if they've done it that well, but they gave everybody restaurant vouchers for a while, to when the restaurants opened up, as a way of subsidizing things and getting you to do that. Now, that didn't work out so well because the virus hasn't gone away. But I think, the reality of getting vaccinated and going there with a mask and, and doing some things that maybe that kind of, maybe we'll be able to evolve into that. I think we're going to have to be a little bit creative and create economic incentives to go with the biological incentives to get us back in track for the kind of economic changes that this is, to recover from.
So I'm going to ask maybe one kind of summary question as we kind of start to wrap, which is, what do you think we've learned about how investment in science has played out for America with this incident, both good and bad, and how might that impact future government support, private support, et cetera?
Well, I think investment in science this has been an amazing return on science. I've also said that leadership makes a difference. I mean, we have the technologies to test everybody all the time. And we've spent that technologies on football teams rather than schools. And, all the athletic facilities and et cetera, all these, so we could have television and advertising and all that stuff and watch sports. And I love the Seahawks. I love sports and all that stuff. But it's like, okay. They're getting it every day. Even the schools are twice a week. I mean, it's like, hey, we can do this. The technology is around to test everybody all the time, whenever you want to talk, whenever you need to do it. And it can make a difference on how things are handled. So I do think that we haven't done things perfectly. I do think technologies have been developed that are being underutilized. I do think that decisions can be made and should have been made about taking over factories and grinding out these machines so that they're available for other than the sports teams. And I'm not happy about it. And then you hear in my voice, I've gone from a little bit, sterner to from, sort of frivolous to this was serious business and it could have been done better. Mm-hmm. And let's hope that we see it.
So with that, I'm going to say thank you. I know this has been an incredibly information dense hour, and I truly appreciate you taking the time with our community. And I can't say enough about the work you do, the work going on with your team and across Fred Hutch. And I think you know how much I support it and really invite everyone else to join me in that. The final thing I would ask, is there anything besides, I've heard you say, wear your masks, make sure you keep them on, stay aware of when vaccinations are available. What else can we do as citizens to really uphold our side of this issue?
Be patient with each other.
Yeah, well, the scarcity thing makes you, becomes an issue. So thank you. And lastly, I just want to, I want to thank everyone who's been with us this whole hour. These have been fantastic questions. I know that we could go on a lot longer, but thank you very much. Thank you everyone for participating in the work that the J does, that Hillel and the J Connect program in specific, and that all the amazing work going on at Fred Hutch. And of course, we invite any of you to learn more, stay connected, and support the work of our three organizations now and always. So thank you. Thank you, Larry, Stephanie, Naomi, wonderful.
Amy, Stephanie, Naomi, been a pleasure. To Simon, to a lot of people, Simon and
yeah, you got a lot of friends on
and
here.
A whole bunch of other people who I recognize on the chat. Thanks for listening in. And
thank you, everybody.
All right.
All right. Thank you.
Bye-bye.
Bye-bye.
And to my family. Bye-bye. Okay.